what there is. So today we are going to talk about regulation of body fluid osmolality, composition and volume. So after a basic introduction, we'll talk about the different body fluid compartments. I think this might have been covered already for you all, but in any case, there are some important things which are pertaining to the renal system. So we'll just go through those as well. Then we'll talk about control of body fluid osmolality, the mechanisms which control the body fluid osmolality, and then the regulation of ECF volume, its concentration, and finally the disturbances of volume and concentration of body fluids. So now the control of body fluid osmolality and the composition and volume, and in fact even the electrolyte balance in body is a concerted function of kidneys, blood, skin, lungs, digestive system, certain hormones and neural mechanisms. So your body fluid osmolality is not simply controlled by kidneys, but all these systems work in tandem to do so. But yes, kidney plays a very important part in these homeostatic mechanisms. So let's learn about the body fluid compartments a little bit. So your total body water, intracellular fluid and extracellular fluid, they form about 60%, 40% and 20% of total body weight. So we call it like a 60-40-20 rule. And the percentage of total body water, okay, is highest in newborns and adult males and lowest in adult females and adults with large amount of adipose tissue. The major cations of your extracellular fluid major cation is sodium, while that of your intracellular fluid is potassium. And major anions of your extracellular compartment are chloride and bicarbonate. In case of intracellular fluid, they are protein and organic phosphates. What are the usual organic phosphates that we think of? Adenosine triphosphate, ATP, adenosine diphosphate, ADP, and adenosine monophosphate, AMP. Now, at your steady state, the major fluid compartments of body, okay, which are the ECF and ICF, they are mostly in osmotic equilibrium, and so they have the same osmolality. Now, this equilibrium is maintained by free water shifts between the extracellular compartment and the intracellular compartment. Okay, this is something that I will tell you in detail towards the end of the chapter. So, a measurement of plasma osmolality, it will provide a measure of both your ECF osmolality also and ICF osmolality. Now, even though the osmolality of both these compartments is quite similar, still there is a marked difference in concentration of electrolytes, okay, between the two, both in case of cations and anions. Your normal plasma osmolality, it ranges from somewhere around 280 to 295 milliosmoles per kg water. Okay, and sodium and its associated ions make the largest contribution, which is around 90% to plasma osmolality. Now, how do we compute the plasma osmolality? So that is done so from the concentration of these ions such as sodium, potassium, urea and glucose, okay, their concentration in millimoles per liter. So plasma osmolality is 2 into that of sodium plus 2 into that of potassium plus that of urea and plus that of glucose. And a factor of 2 is used for sodium and potassium ions to account for the associated anion concentration. Now, since sodium is the most predominant contributor of osmolality in the plasma, so if you simplify this calculation, then it comes out to be, and of course, presuming that plasma glucose and urea are in normal range, then this comes out to be plasma osmolality in millimoles per kg equal to 2 into that of plasma sodium. Okay, again, always in millimoles per liter. Now, sodium and its associated ions, okay, mainly chloride, they are usually confined to your extracellular fluid. So, if there is retention in the extracellular fluid, it will directly relate to osmotic effect of these two ions. Which ones? Sodium and chloride. So, the amount of sodium in extracellular fluid will ultimately determine its volume. So whenever we evaluate abnormal sodium levels in the plasma, it's usually very tempting to suspect in sodium balance, but mostly what comes out to be is not sodium alteration, but an issue of water balance. Okay, so that's something that's better kept in mind when we look at this in a clinical scenario. Now let's talk about water balance in body. Okay, so how is the water input to the body done? Through body fluid. Water is added to the body fluids by one, of course, ingestion of water in form of fluid and constituents of foodstuffs. So water intake is quite variable. It might range from 0.5 to 2 liters per day, depending on one's social, personal, environmental conditions or social personal habits. And yes, we know already that generally people living in hot climate drink more water. Also, ingestion of water is mainly controlled by the thirst center, right? And the usual mechanism is that if there is increase in plasma osmolality, then your thirst center will get stimulated and promote water ingestion. And the other method of input of water is through endogenous production during oxidation of foodstuffs. That roughly adds about 300 ml of water to the fluids per day. Now, water output. So, a variable amount of water is lost from the body in form of urine, feces, sweat, and even insensible loss. Okay. So, what is insensible loss of water? Insensible as a word here implies what the individual is unaware. Okay. So this insensible loss of water, it occurs by evaporation from cells of skin and respiratory passages. Okay. Then there is water loss in sweat. So water loss by sweat from skin can vary from 100 ml per day in routine at room temperature, okay, of 23 degrees to around 1400 ml in hot weather to even 5000 ml following prolonged exercise. Okay, then there is loss in feces also, which is around 200 ml per day is lost in a healthy individual through feces, and most of the water that enters GIT is reabsorbed by the intestines. Then the fecal loss of water is tremendously increased in diarrhea, right? And GIT water loss can also occur in case of vomiting. And then finally, water loss in urine. So roughly 1500 ml of water is eliminated from the body in urine. And just know that water losses in kidney are quite variable. Okay, so it's also important to know that the water loss in the sweat, feces, and evaporation from lungs and skin is not as well regulated. Okay. Now, the renal loss that occurs of water is variable, but unlike the other systems which I just mentioned, this is very well regulated to maintain the water balance. So, for water balance, water output is precisely matched with water intake by the kidneys. Okay, so kidneys produce a small amount of concentrated urine. Okay, that means a hyperosmotic urine with respect to plasma when the intake is low or if the losses are more, right? And conversely, the kidneys produce a large amount of dilute urine, which means hypoosmotic with respect to plasma when the water intake is high. So in a normal individual, basically depending pretty much on ADH concentration, the urine osmolality can vary from 50 to 1200 milliosmoles per kg water. Okay, and with a urine volume of 18 to 0.5 liters per day. Okay, you have all the values here just for your reference. Then let's learn about the disorders of water balance, okay, which alter body fluid osmolality. So, the changes in body fluid osmolality usually occur due to change in plasma sodium. So, in that case, you have a positive water balance. What does that mean? That the intake is more than excretion. That results in a decrease in body fluid osmolality and hyponatremia, right? And then you can have a negative water balance, which means the intake is less than excretion. That would result in an increased body fluid osmolality and hypernatremia. Okay, so what are the factors which control water balance? So now the input of water, what I mean is thirst, and output of water, that means urinary excretion, they are both controlled by plasma osmolality and your blood volume. So plasma osmolality controls water balance by stimulating the thirst center and ADH secretion. Okay, these two topics are quite important in this unit. Okay, we'll of course go through this again shortly. So just keep your ears open for the thirst mechanism and the role of ADH. So significant changes in ADH secretion occur by a small change, which is roughly even 2% change in the plasma osmolality can have significant changes in your ADH secretion. Okay. And then in case of blood volume, so normally the water balance is regulated through osmoreceptors. Okay. But a significant decrease in blood volume, I mean more than 10%, also stimulates thirst and ADH secretion from posterior pituitary. Okay. So if your blood volume is markedly decreased, then ADH release is stimulated even if plasma osmolality is low. Okay. And decreased blood volume is sensed by low pressure receptors in the atria and pulmonary vessels. So both these factors are very important for controlling your water balance, both plasma osmolality and blood volume. Now, what are the mechanisms controlling the blood fluid osmolality? So this is what I was just referring to. So the main mechanisms regulating your body fluid osmolality, we'll just come to those. So now the control of body fluid osmolality, meaning the defense of tonicity of extracellular fluid, is primarily a function of vasopressin secretion and thirst mechanisms, which I just referred to. Okay, so when the effective osmotic pressure of plasma rises, okay, the osmoreceptors located in the anterior hypothalamus, they get stimulated. Okay, and stimulation of receptors results because of their shrinkage caused by cellular dehydration. Okay, how would the cellular dehydration occur in the first place? Either because of a deficiency of total body water, right, or because of excessive intake of sodium chloride. which will cause the water to shift from your intracellular compartment to the extracellular compartment. So now these stimulated osmoreceptors, what will they do in turn? Okay, so let's go back here. First, what happened? The effective osmotic pressure rose. Okay, your osmolality increased. Okay, in the ECF, and then that caused the stimulation of osmoreceptors. Okay, and what happened cellularly to cause the stimulation of osmoreceptors? Basically, at cellular level, the receptors had shrunk. Okay, they shrunk because of cellular dehydration, and that shrinking caused the stimulation of osmoreceptors in your anterior hypothalamus. Now, once these are stimulated, then what happens? Okay, two things. So one, increased thirst, okay, which regulates your water intake. All right, and what is the other thing that happens when the osmoreceptors are stimulated? Is increase in your ADH secretion or vasopressin secretion here. Okay, and that will regulate your water excretion or retention, whichever, by the kidneys. Okay, I hope this is clear. Now, when the reverse happens, okay, that the effective osmotic pressure of plasma decreases, okay, then plasma, what that means, of course, is when plasma becomes hypotonic, then your vasopressin secretion will decrease, okay, which means that water excretion will increase in excess of the solute, okay, and thirst will also decrease, which means that the water intake itself will decrease. So I think that's an absolute reversal of what we just read. Okay, so once you understand one of these, you of course know what happens when the converse situation arises, right? Now, the main mechanisms which are involved in regulating body fluid osmolality are: one, your role of ADH hormone, okay, role of thirst mechanism, and the renal mechanisms for urine dilution and concentration. So this third one is what we went through, what the entire last unit was about. Okay, so that's that. And this is your thirst mechanism, which we just spoke about. And role of ADH hormone is something we spoke about last time also. I thought we can go through it again today, just for reinforcement. And I would suggest that you add the information about ADH from your what you learn in your endocrinology lecture also, which is related to the kidney or control of plasma osmolality to this. So role of antidiuretic hormone, yes, we know it prevents diuresis, chiefly concerned with conservation of water. And why is it called vasopressin? I had told you last time, because it causes vasoconstriction, also called as arginine vasopressin. Okay, and we know its action on kidney by now, which is regulation of water balance, right, where it can decrease the excretion of free water, right, and that's why it's called a that is an antidiuretic and concentrating effect, hence its name. Okay, the site I had told you, they line the DCT and collecting duct of renal nephron, and it increases permeability of these cells to water. Then we had also talked about the mechanism of action. So I'm reiterating this one more time for you. So it exerts its antidiuretic effect by binding to specific plasma membrane receptor, which is called as the V2 receptor on the basal or the capillary side of cell, where it activates adenyl cyclase. The increase in intracellular CAMP activates protein kinase on the opposite side, the apical or the luminal side of the cell. The activated protein kinase leads to rapid insertion of protein water channels called as aquaporins in plasma membranes of principal cells of collecting duct. Okay, so remember the main role is in collecting duct. And then through these protein water channels, the aquaporins, the water rapidly moves from tubular lumen into the collecting duct cells. So I had told you last time also, aquaporins is an important question, both as a short note and even, one among the favorite questions asked in Vivas also. Okay. So aquaporins, I told you, are of different types. 1, 2, 3 found in kidneys, 4 in brain, 5 in salivary, lacrimal glands, and in the respiratory tract. Now your ADH responsive aquaporin is aquaporin 2. Okay, and that is stored in the endosomes inside the cells. It is translocated by ADH into the luminal membrane of, again, principal cells. Sorry for that misspelling there. Principal cells of collecting duct. Okay, and then, as I said, through these protein water channels, the water rapidly moves from tubular lumen into the cells of collecting duct. From inside cells, water moves into the ECF then through aquaporin 3 and 4, which get inserted into the basolateral membrane of cells. Okay, so first is the role of aquaporin 2, okay, which is translocated into the luminal membrane of the principal cells. And then through this, the water will move from tubular lumen into the cells of collecting duct. Now from inside of those cells, water will now move to the ECF, and that will occur through aquaporin 3 and 4, which is in the basolateral membrane. So this one was in the luminal membrane, okay, or the apical one, and this is now in the basolateral membrane. So broadly, this is what is happening, and this is your mechanism, that what gets activated and which protein and how this process occurs. Now the regulation of extracellular fluid, it is primarily mediated through regulation of amount of osmotically active solute present in it. Okay, and we already know that major solutes of ECF are the salts of sodium, and out of all these salts, sodium chloride is the most abundant one. Now, since kidneys are a major route of sodium chloride excretion from body, so of course they play a very important role in its regulation of the extracellular fluid. Now, kidneys get signals from their volume sensing system to make the desired adjustment in sodium chloride excretion. Okay, and your volume sensors, they generate signals in response to changes in effective circulating volume, your ECV. Okay, and volume sensing signals, then they control volume of ECF by controlling renal excretion of sodium chloride. Okay, so you have a volume sensing system basically, which senses changes in mainly sodium chloride concentration. Okay, then it generates signals in response to that change of the effective circulating volume. Okay, and then that will control the renal excretion of sodium chloride and water. So that's how the entire system is regulated. Okay, and so when we talk about this process of ECF volume regulation, we talk about effective circulating volume and volume sensing system, as I just mentioned, and volume sensor signals, which are neural and hormonal, which constitute the neural and hormonal control of sodium chloride excretion and sodium balance. Okay. Now about the ECF and volume sensors. So portion of so in a physiological setting, the ECF refers to the portion of ECF volume that is present in the arterial system, okay, under particular pressure, and is effectively perfusing the tissues. Okay, and about 0.7 liter of the vascular volume, which is 20% of plasma, 5% of ECF, or 1.7% of total body water or 1% of body weight. That is what forms the ECV, effective circulating volume. Now, this ECV is regulated by the volume sensors, which I just mentioned, which are located entirely within the vascular tree. Okay, and sensation of ECV is related to fullness. That means volume and pressure in the vascular tree. Now, in a normal state, okay, the variations in ECV are reflected as parallel variation in the ECF volume and also in the vascular volume, in your blood pressure and cardiac output. Okay, what that means is that if there is a decrease in ECF, vascular volume, or your BP or your cardiac output, it will be sensed in the body as a decrease in your ECV. Okay, now how is this ECV maintained? Okay, so the maintenance of ECV and sodium balance are quite closely related. So sodium loading will produce expansion of ECV and sodium loss will lead to depletion of ECV. Okay, what that means is when ECV is decreased, renal sodium chloride excretion is reduced. Okay, so this adaptive response, it will restore your ECV to normal and in this way maintain adequate tissue perfusion. Conversely, what will happen if there is increase in ECV? That will of course result in an enhanced renal sodium chloride excretion, which we also call as natriuresis. And again, this is also an adaptive response to restore the ECV to its normal point. Now, what are these various volume sensors which we just talked about? Okay, so the receptors which are located in the vascular system, okay, and respond to a degree of stretch of the vessel wall, okay, and not to the volume of the vessel. These are the volume sensors or volume receptors that I just spoke about, also called as baroreceptors. Okay, so again, they are in the vascular system, they respond to the degree of stretch of vessel wall, but not to the volume of the vessel. Remember that. Okay, now the various volume receptors, volume sensors are: one, you have your vascular volume receptors, and then you have mainly your hepatic sensors. So among the vascular volume receptors, you have two categories: low pressure volume receptors and high pressure volume receptors. The low pressure volume receptors are located in cardiac atria and pulmonary vasculature. The high pressure volume receptors are located in carotid sinus, aortic arch, and juxtaglomerular apparatus of kidney. We know that, we've already done that in the first unit. And in case of hepatic sensors, even though they are not as important as the vascular volume receptors, but they also have a role in regulating your renal sodium chloride excretion by reflexly regulating your renal sympathetic discharge. That's how the hepatic sensors work. And they also are involved in the regulation of GIT, sodium absorption. Okay, so for example, if the sodium concentration of a portal blood vein is increased, there is reflex reduction in the jejunal sodium chloride absorption. Okay, so these are your main two: vascular volume receptors and hepatic sensors. Now let's go on to the neural regulation. So your sympathetic nerve fibers, they innervate afferent arterioles and efferent arterioles of glomerulus as well as nephron cells. Okay, so the renal sympathetic stimulation is induced by vascular low and high volume sensors in conditions of negative sodium balance, which I just told you earlier, which means decreased ECV, leading to decrease in sodium chloride excretion by certain mechanisms. What are they? So one is reduction in GFR due to vasoconstriction of afferent arterioles. This is induced by sympathetic stimulation. Okay, we have done this, so try to recall how the sympathetic stimulation occurs whenever there is reduction in GFR due to vasoconstriction. Now, this reduced GFR, it leads to decrease in filtered load. What is filtered load? Filtered load we had done last time is the product of GFR and plasma sodium concentration. Okay, so now reduction in filtered load, this helps in sodium conservation. But the changes in filtered load of sodium are not reflected in parallel changes in urinary sodium excretion. Why? Because you have a glomerular tubular balance and a tuberglomerular feedback, don't you? You have an auto-regulation mechanism in place, right? So whatever reduction in GFR is happening, it is handled at the level of changes in your filtered load, okay? And not so much by change in urinary sodium excretion, because you have an auto-regulation system in place for that. Then what is the second mechanism? Second is increased sodium reabsorption along the nephron, okay? And that is produced by stimulation of alpha adrenergic receptors. And your proximal tubule is the most important segment, which is influenced in this case by the sympathetic stimulation. The third mechanism is stimulation of renin secretion from cells of afferent arterioles. That is produced by activation of beta adrenergic receptors, okay? And that results in an increased plasma concentration of angiotensin II and aldosterone. Okay, so this was about renal sympathetic stimulation. Now coming to renal sympathetic inhibition. So that would be induced by vascular low and high pressure volume receptors in condition of positive sodium balance, which means increased ECV. That would lead to increased sodium chloride excretion by the reverse of what we just spoke about. Then you have your hormonal regulation. Okay, so I'm sure you already know about the RAA system, renin angiotensin aldosterone system. And I'm sure you would have or will read about that in detail in your endocrinology. So this system, when stimulated with volume depletion, okay, it results in decreased sodium chloride excretion. And conversely, when suppressed with volume expansion, increases sodium chloride excretion. So your renin is secreted by smooth muscles of afferent arterioles of kidney, right? We know that. And what are the stimuli for renin secretion? So one is your reduced perfusion pressure, other is your increased renal sympathetic discharge, and third is your decreased delivery of sodium chloride, which is sensed by macula densa cells. I remember we have talked about this in a few diagrams also. So let's see what happens here. Okay. Do we know the conversion of renin into angiotensin II? So basically, your tensinogen is produced by liver, okay, which is converted into angiotensin 1 by renin, okay, secreted by the kidney. Then this angiotensin 1 is acted by your angiotensin converting enzyme from the lungs into angiotensin 2. All right. And it is your angiotensin 2 which has the main physiological role to play, okay, in this case. So what does this do? Angiotensin 1, it will stimulate aldosterone secretion by the adrenal cortex. That is the first thing angiotensin will do. Okay, then what will it do? It will increase blood pressure by arteriolar vasoconstriction. That is the second thing. Then it will stimulate, it will stimulate ADH secretion, okay, and thirst. All right. And it will enhance, now it will directly enhance the sodium chloride reabsorption by the proximal tubules. Okay, so this is your mechanism of decreased sodium excretion. Okay, so as a result of the role of angiotensin 2 on all these, whether it's your brain or the renal cortex or the kidney itself or the effect on blood pressure, what is the end result? The sodium excretion and the water excretion is reduced. Okay. Now, there is also role of ANP. Before that, let's go to the role of aldosterone. So aldosterone also enhances sodium chloride reabsorption from the thick ascending limb of loop of Henle. And how does it do that? By two mechanisms. Now, this is a diagram that we had gone through earlier in Unit 2, which I'm reiterating again here. So, aldosterone, it enhances sodium chloride concentration in the thick limb of loop of Henle, okay? And by its action on one, you're the sodium potassium chloride symporter, which is present at the apical membrane, and two, its action on the sodium potassium ATPase on the basolateral membrane. So on both these mechanisms, you have aldosterone acting, and both these will result in sodium chloride reabsorption in the thick ascending limb of loop of Henle. And yes, we already know about the third factor, which is antidiuretic hormone, right? We just spoke about that, which is secreted by your posterior pituitary, okay, which increases because of volume depletion or decrease in effective circulating volume. That leads to water retention by kidneys and re-establishing of euvolemia. And the reverse will happen in volume expansion. Then another factor is your atrial natriuretic peptide, or ANP. Okay, so ANP is released from atrial myocytes by the atrial stretch caused by volume expansion, increased ECV, and its actions are opposite to that of renin. Okay, so that's about ANP. We will come across this in a diagram very shortly. Okay, so as I said, its actions are opposite to that of the renin-angiotensin-aldosterone system, and that results in an increased urinary excretion of sodium chloride. Now, how does that occur? So basically, ANP causes increase of GFR by vasodilation of afferent arteriole and vasoconstriction of efferent arteriole. Then it also causes inhibition of renin secretion by afferent arterioles. It causes inhibition of aldosterone secretion, okay, by inhibiting renin secretion, and also by direct effect on adrenal cortex cells. Also causes inhibition of ADH secretion by posterior pituitary and inhibition of ADH action on the collecting duct, inhibition of sodium chloride reabsorption by collecting duct, by inhibiting aldosterone secretion, by inhibiting ADH secretion, and direct effect on collecting duct cells. So you see, basically, when there is volume expansion because of increased effective circulating volume, that will cause a atrial stretch of the atrial myocytes and release and cause a release of ANP from the atrial myocytes. And now this ANP is working opposite to the RA system and will increase the urinary secretion of sodium chloride. And mostly, apart from increasing the GFR, it is mostly having an inhibitory effect on all the systems, whether it is renin secretion by afferent arteriole, whether it is aldosterone secretion, whether it is renin secretion, whether it is inhibition of ADH by pituitary, inhibition of ADH action on collecting duct cells. Okay, so directly and indirectly, it is inhibiting all of these other mechanisms. But yes, it is increasing GFR by vasodilation of afferent arteriole and vasoconstriction of efferent arteriole. So this is what I was just referring to. Just have a look, just go through this diagram. Can you relate what we just spoke about? So one is a situation where it is showing regulation of sodium excretion, okay, with volume expansion, okay, which is the one shown in the diagram for now. So volume expansion, which means like a large increase in ECF volume and ECV. When do they occur? They can occur following excessive salt intake or in case there is excessive administration of IV saline, right? Now, this increase in volume expansion is detected by vascular volume receptors that we spoke about earlier, and that initiates a response that ultimately leads to sodium excretion. Okay. Now, what is the mechanism of increased sodium excretion? Okay, so one is your decreased renal sympathetic discharge. So here, if you see, volume expansion has happened and there is decreased sympathetic discharge. Then there is suppression of your renal angiotensin aldosterone system. Okay, here. Then there is increased in ANP secretion, which we just spoke about from the atrial myocytes. Okay, why did that happen? Because they got stretched. See here, because the atrial myocytes, they went through an atrial stretch caused by volume expansion or the ECV increase, right? So there is increased ANP secretion, okay? And then, I just told you about all the functions of ANP where it plays a major inhibitory role on most of the systems, most importantly being on causing ADH suppression. And we also just read about inhibiting ADH action on collecting duct, ADH secretion itself in the posterior pituitary. Even the aldosterone secretion is being inhibited, this one which we just spoke about. So it has a collectively inhibitory effect on most of the systems other than increasing GFR. Okay, so the renal response to this entire system, okay, is what? As I said, increased GFR will be there, okay? Then there'll be reduced sodium absorption in the PCT, okay? There'll be increased delivery of sodium chloride to the beginning of collecting duct, okay? And there'll be decreased sodium reabsorption in the collecting duct. So these are the renal responses to this mechanism. Okay, ultimately what happened? There is more of sodium and water excretion, right? And the important difference between a situation of euvolemia and volume expansion is that the renal response in case of volume expansion involves the entire nephron. But in case of eovolemia, the renal response mainly involves the collecting duct. Okay. Now, what is the second situation here? So now sodium excretion with volume contraction. So I'm sure you can come up with a converse diagram of this entire process. So in that case, when there is volume contraction, the sympathetic activity will increase. The renin angiotensin aldosterone mechanism will increase. Okay. ANP secretion from atrial myocytes will decrease, and there will be stimulation of ADH release. So now, what do you think will be the renal response to all this, to this particular converse mechanism? So, of course, in this case, the GFR will decrease. And yes, exact opposite. We had said earlier that sodium reabsorption is reduced in PCT. In this case, sodium reabsorption in PCT will increase. Okay. Previously, we had said there'll be increased delivery of sodium chloride to the beginning of collecting duct. So now there'll be decreased delivery of sodium chloride to the beginning of collecting duct. Okay. Then we had said previously that sodium reabsorption in the collecting duct will be reduced. Okay. And here, of course, sodium reabsorption in the collecting duct will increase. Okay. So, as I said, in case of eovolemia, the effects are limited to the collecting duct, okay? But in case of volume contraction or expansion, as we spoke about earlier, the effects are reflected in the entire nephron, okay? I hope this is somewhat clear. So this is a summary, this table, just have a look. This is a summary of the neural and hormonal regulation of sodium chloride excretion that we just spoke about. So here, for your neuronal control, sympathetic activity decreases sodium chloride excretion by these three mechanisms: decreased GFR, increased renin, increased tubular sodium chloride reabsorption. In case of hormonal control, we spoke about three systems, right? The increased activity of RAAS decreases sodium chloride excretion by, I just told you, increased proximal tubule absorption, increased ADH secretion by angiotensin 2, and increased tubular reabsorption of aldosterone. So these three we spoke about, the RA system, the ANP, and increased ADH. ANP will increase sodium chloride excretion by increasing GFR. Rest are all inhibitory effects: decreased renin, decreased aldosterone secretion, decreased sodium chloride, decreased water reabsorption by collecting duct, decreased ADH secretion and action of ADH in collecting duct. And the third mechanism in hormonal control is increased ADH secretion, which decreases water excretion. How? By increasing the water absorption by the collecting duct. And I just told you the mechanism of how ADH does that through the role of aquaporins. Now, what are the disturbances of volume and concentration of body fluids? So, we all know what is dehydration, okay, right? We know that is the volume or the fluid loss. And then we hear of overhydration, which is fluid gain. And depending on the osmolality relative to the plasma, the fluid may be in of three types. It may be isotonic fluid. What does that mean? So fluid having the same osmolality as that of plasma, right? And you must have these examples on tips, whether for your physiology exam or even otherwise. Clinically, you need to know these. that which fluids have same osmolality as plasma: 0.9 sodium chloride, 5% glucose, 10% mannitol, and 20% urea. And these solutions, they don't change the volume of body cells, right? What is a hypotonic fluid? A fluid with osmolality less than that of plasma. Such solutions, they cause the cells to swell, and if sufficiently dilute, to even burst or lyse. And hypertonic fluid is the fluid having osmolality more than that of plasma, and such solutions cause the cells to shrink, that means to undergo crenation. Now there are certain disturbances of fluid volume and concentration. So what are those situations that can arise? So you can have an isoosmotic fluid volume expansion or a contraction. You can have a hyperosmotic volume expansion or a contraction, or you can have a hypoosmotic volume expansion and contraction. So this is the last main topic for today. I need you to pay attention to this diagram for understanding this. And whatever I tell you at the end, I will show you a table which will compile all that I'm explaining shortly. Okay, so don't worry about it. If you can just have this table in mind, you will, I'm sure, remember this. So this diagram is called as Darrow-Yannette diagram. Okay, it's given in Genong. And so let's learn about the disturbances of fluid volume now. So your first situation that we are looking at here is an isoosmotic volume expansion. Okay, what do you think can cause a isoosmotic volume expansion? So possibly by infusion of isotonic fluids. I just told you which are the isotonic fluids. For example, a 0.9% sodium chloride solution. Okay, now on doing so, what are the consequences? So your ECF volume, it increases. Okay, so just see that here this axis is showing osmolality and your x-axis is showing volume. So the first consequence is that your ECF volume will increase in case of volume expansion, or let's say infusion of an isotonic fluid. Okay, but the ECF osmolality does not change. Okay, so ECF volume बढ़ रहा है, but osmolality वही है. And so water does not shift between the ECF and ICF compartments. But what happens? Plasma protein concentration decreases because of dilution effect of additional fluid, right? क्योंकि volume तो बड़ा है. That results in decreased plasma colloid osmotic pressure. क्यों? क्योंकि प्लाज्मा प्रोटीन्स की ही concentration कम हो गई है, तो प्लाज्मा कोलॉइड ऑस्मोटिक प्रेशर भी कम हो जाएगा, right? And because of this decreased colloid osmotic pressure, water moves out of blood vessels and gets distributed in the interstitial compartment. ठीक है? हेमाटोक्रिट में क्या होगा इफेक्ट? हेमाटोक्रिट कम हो जाएगा because see your fluid has gotten added in the ECF, है ना? And this will cause dilution of RBCs. But हमने शुरू में ही कहा था कि ECF की osmolality तो change नहीं हो रही है. So the RBCs will neither shrink nor swell. ठीक है? They will be diluted. Hematocrit will decrease, but they will not shrink or swell. And your arterial blood pressure will increase. क्यों? क्योंकि ECF volume बढ़ा है तो आपका arterial blood pressure भी बढ़ेगा. So these will be the consequences of an isoosmotic expansion. और उसका corrective response क्या होगा body में? तो corrective response ये होगा कि the the change that has occurred in the plasma volume will be sensed by the vascular volume receptors. They will bring about excretion of large volumes of hypotonic urine, which we also call as water diuresis. I had talked about it in the last lecture. And that will gradually restore the plasma volume and osmolality to normal. ठीक है? Now, do we know something about isoosmotic volume expansion? I hope we do. Now, let's go on to the next situation. This one. So, now let's talk about a isoosmotic volume contraction. Okay. So, as you can see, the difference between these two. So, first of all, how will the isoosmotic contraction occur? Because of course, loss of isotonic fluid, which occurs in which conditions? Diarrhea, vomiting, hemorrhage, ascites, burns, right? This is how isoosmotic fluid is lost. Now, coming to consequences of an isoosmotic volume contraction. So, as you can see clearly the difference between these two, here the ECF volume is decreased. Again, ECF osmolality does not change as happened in this case. And again, similar to this case, the water doesn't shift between the ECF and ICF compartments. Now, reverse effect will happen. I think that's easy to see. That इस केस में प्लाज्मा प्रोटीन्स का क्या होगा? यहाँ पे वो कम हो गए थे because of dilution by the additional fluid. तो यहाँ पे वो बढ़ जाएंगे because of loss of ECF. ठीक है, loss of ECF volume से plasma proteins will end up getting concentrated. ठीक है. Now, because of plasma proteins, okay, the plasma volume is reduced less as compared to the interstitial fluid. Why? Because plasma proteins, they tend to hold your fluids. ठीक है. So, जब plasma protein concentration बढ़ गई, तो plasma volume की कमी कम होगी as compared to the interstitial fluid. ठीक है. हेमाटोक्रिट का क्या होगा? उल्टा हो जाएगा. हेमाटोक्रिट पहले यहाँ पे कम हो गया था, राइट? क्योंकि डाइल्यूशन हो गई थी आरबीसीज की. यहाँ पे क्या होगा? बढ़ जाएगा, राइट? बिकॉज ECF लॉस से आरबीसीज अब नाउ दे विल गेट कॉन्सेंट्रेटेड. ठीक है. एंड हमने पहले ही कह दिया कि ECF ऑस्मोलैलिटी दोनों सिचुएशंस में चेंज नहीं हो रही है. सो इन बोथ केसेस RBCs विल नाइदर श्रिंक नॉर स्वेल. ठीक है. इस केस में हमने क्या कहा था ब्लड प्रेशर के बारे में? ब्लड प्रेशर बढ़ जाएगा बिकॉज वॉल्यूम, ECF वॉल्यूम आपका बढ़ गया है. ये देखो इस एक्सिस पे. और इस केस में आप ब्लड प्रेशर क्या होगा? कम हो जाएगा बिकॉज ECF वॉल्यूम ही कम हो गया. ठीक है. अब इसका करेक्टिव रिस्पॉन्स क्या होगा? सो अगेन, अब प्लाज्मा वॉल्यूम कम हो गया है तो दिस विल इनहिबिट द vascular volume sensors. ठीक है. And reflexly restore the plasma volume by decreasing sodium and water excretion. ठीक है. One important thing to remember is that जो thirst produce होती है because of volume receptors, it is quenched with isotonic salt solution instead of plain water. Okay. So, are we clear about these two situations of isoosmotic volume expansion and contraction? I hope we are. So we can proceed to then what happens in case of hyperosmotic volume expansion. ये कब होती है? In a situation when there is excessive amount of hypertonic saline administered. Okay. So, अब हमारा इफेक्ट ऑन ऑस्मोलैलिटी आना शुरू हो जाएगा. ठीक है. यहां देखो ऑस्मोलैलिटी वाई एक्सिस पे आपकी कांस्टेंट थी, है ना. अब आप देखो आपकी ऑस्मोलैलिटी में भी चेंज आ रहा है. वॉल्यूम में भी चेंज आया, वॉल्यूम में तो आ ही रहा है बट ऑस्मोलैलिटी में भी चेंज आ रहा है. सो इस केस में हाइपर ऑस्मोटिक वॉल्यूम एक्सपेंशन में योर ECF osmolality is increased. ऐसा क्यों हुआ? बिकॉज सोडियम क्लोराइड ऑस्मोल्स हैव बीन एडेड टू दी ECF. ठीक है, हमने हाइपरटोनिक सलाइन दिया ना, इसलिए. तो अब क्या कॉन्सिक्वेंसेस होंगे? अब आपका वाटर विल शिफ्ट फ्रॉम ICF टू ECF. ठीक है, इन दिस केस यहाँ पे कोई शिफ्टिंग नहीं हुई थी बिटवीन ICF ECF, और ना ही RBCs की कोई श्रिंकेज या स्वेलिंग हुई. ठीक है, इस केस में अब वाटर विल शिफ्ट फ्रॉम ICF टू ECF, और इस वजह से ICF ऑस्मोलैलिटी बढ़ जाती है टिल इट इक्वल्स टू दैट ऑफ ECF. ठीक है, अब ECF वॉल्यूम जो है वो भी बढ़ गया अगर आप देखो यहाँ पे, बिकॉज यू हैव डन फ्लूइड एडिशन एज वेल एज आपने भी फ्लूइड दिया है जो एक्सेसिवली हाइपरटोनिक सलाइन दिया है, और प्लस फ्लूइड ICF से भी ऐड हुआ है, राइट? सो आपका ECF वॉल्यूम बढ़ जाएगा, और ICF वॉल्यूम ऑफ कोर्स कम हो गया बिकॉज यहां से फ्लूइड शिफ्ट हो गया ECF में, राइट? अब प्लाज्मा प्रोटीन में क्या होगा? तो सबसे पहली सिचुएशन वाला केस कि प्लाज्मा प्रोटीन्स कम हो जाएंगे बिकॉज ऑफ इंक्रीज इन ECF वॉल्यूम. ठीक है, तो प्लाज्मा प्रोटीन की पे क्या इम्पैक्ट होगा वो आप रिलेट करो रिलेट दैट टू द चेंज इन ECF वॉल्यूम. अगर ECF वॉल्यूम में चेंज हुआ है तो प्लाज्मा प्रोटीन में भी चेंज होगा. अगर ECF वॉल्यूम बढ़ता है जब भी किसी भी सिनेरियो में, जैसे कि इसमें भी बढ़ा था आइसोस्मोटिक में भी, तो प्लाज्मा प्रोटीन हमेशा फॉल कर जाएगा. ठीक है, एंड सिमिलरली हेमाटोक्रिट कम हो जाएगा अगेन बिकॉज ऑफ इंक्रीज इन ECF वॉल्यूम. क्यों ऐसा क्यों हुआ था? क्योंकि addition of fluid of ECF dilutes RBCs, right? But in this case, RBCs will shrink. ठीक है, क्यों? क्योंकि ECF osmolality बढ़ गई है, राइट? वो हमने शुरू में ही कह दिया था. तो चेंज अब आ गया RBCs में मेनली, बाकी मोस्टली चीजें osmolality बढ़ गई और RBC में चेंज आ गया, श्रिंक हो गए. Otherwise, the situation is very similar to your this one, isn't it? प्लाज्मा प्रोटीन्स, हेमाटोक्रिट, ये सब में, और इवन इनफैक्ट आपके ब्लड प्रेशर भी सिमिलर कॉन्सिक्वेंस है. इधर भी ब्लड प्रेशर बढ़ रहा है क्योंकि ECF वॉल्यूम बढ़ गया. तो बिकॉज आपका ये फैक्टर दोनों में कॉमन है, काफी चीजें सिमिलर हैं. फर्क क्या आया इसमें, इन दो कंपार्टमेंट्स में? यहाँ ऑस्मोलैलिटी बढ़ गई तो उससे हमारे आपके RBCs पे इफेक्ट आ गया, RBCs श्रिंक हो गए. और दूसरा क्या हुआ? ऑफ कोर्स यहां से शिफ्टिंग हो गई ICF से ECF में. ठीक है, ये है मेन डिफरेंस. अब इस केस में करेक्टिव रिस्पॉन्स क्या होगा? अब प्लाज्मा ऑस्मोलैरिटी बढ़ गई थी, दिस विल प्रमोट वाटर रिटेंशन. ठीक है, while an increase in plasma volume inhibits the same. So ultimately क्या होता है? वॉल्यूम overrides tonicity. ठीक है, so increased volume will suppress thirst and ADH, leading to excretion of large amount of hypotonic urine. ठीक है, तो large amount of hypotonic urine excrete हो गया, so that will bring down the plasma volume. ठीक है, and the natriuretic hormones which are secreted only when there is change in osmolality, not volume change. Okay, those natriuretic hormones will promote sodium excretion and correct the osmolality. Okay, I hope you all are getting some sort of clarity for this. Okay, कि जब भी प्लाज्मा osmolality बढ़ रही है, it will promote water retention. जबकि प्लाज्मा वॉल्यूम में जब जब इंक्रीज आता है वो इनहिबिट करता है, बट इस सिचुएशन में द इफेक्ट्स ऑफ वॉल्यूम दे ओवरराइड द इफेक्ट्स ऑफ टॉनिसिटी। ठीक है, तो अब हम वॉल्यूम की बात करते हैं। प्लाज्मा वॉल्यूम तो बढ़ गया है, उसने आपका ये सारा जो था एक सेकेंड, हाँ, उसने आपका इन दोनों पे इफेक्ट कर दिया। ठीक है, थर्स्ट को सप्रेस कर दिया, राइट, एंड एडीएच को भी सप्रेस कर दिया। ठीक है, तो आपकी वाटर इनटेक कम हो गई, जो इसका रिवर्स मैकेनिज्म हुआ है, और एक्सक्रीशन ऑफ, उल्टा हो गया, एक्सक्रीशन ऑफ लार्ज अमाउंट ऑफ हाइपोटोनिक यूरिन हो गई, उससे आपका प्लाज्मा वॉल्यूम कम हो गया। ठीक है, और आपकी नेट्रियूरेटिक हॉर्मोन्स तभी सिक्रीट होते हैं जब प्लाज्मा ऑस्मोलालिटी में चेंज होता है, नॉट वॉल्यूम में, जो कि हुआ है इस केस में, है ना। तो उससे दैट दोज प्रमोट सोडियम एक्सक्रीशन और आपकी ऑस्मोलालिटी में जो भी इशू है करेक्ट करते हैं। ठीक है, shall we go on to the next one? So now come to hyperosmotic volume contraction. So I hope अब आप खुद भी यू कैन फिगर आउट, है ना, क्या होना चाहिए इस केस में। पहले तो ये होती क्यों है? दिस मेनली हैपेंस ड्यू टू, अगर वाटर इनटेक ही कम है, या डायबिटीज मेलाइटिस है, या डायबिटीज इंसिपिडिस है, if one is in a desert environment and excessive sweating happens, alcoholism में होती है, and even in tracheostomy patients जहाँ पे insensible loss of water can go up to 500 ml, okay, from the lungs. So इस केस में क्या होगा? अब आप देखो, just compare these two, that there is, यहाँ पे as you can see ECF volume is reduced. Why? Because water loss has happened in all these situations which I mentioned, right? ECF osmolality is increased, right? Because again water is lost. ठीक है, so again your water shifts from ICF to ECF. ठीक है, पहले भी अभी यही हुआ था, राइट? ECF osmolality इसमें भी बढ़ी थी। इसमें क्यों बढ़ी थी? बिकॉज हमने हाइपरटोनिक सलाइन दिया था। इसमें क्यों बढ़ गई? बिकॉज इसमें क्यों बढ़ गई? बिकॉज वाटर लॉस हो गया। सो वही सिचुएशन है कि यहाँ से अब ICF से वाटर शिफ्ट करेगा ECF में। ठीक है, इस शिफ्ट से अगेन ICF ऑस्मोलैलिटी बढ़ जाएगी, जो इधर भी यही हुआ था, राइट, until it equals that of ECF. Same, exactly same until now. And the ICF volume, it decreases because of shift of water. ये भी सेम है। ठीक है, प्लाज्मा प्रोटीन में क्या होगा? अब यहां पे चेंज है। ठीक है, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन बढ़ जाएगी। क्यों बढ़ जाएगी? बिकॉज इस केस में डिक्रीज इन ECF वॉल्यूम है। ठीक है, दिस इज वेयर द चेंज कम्स। और ये हमने पहले भी किया था, राइट? आइसोऑस्मोटिक में हमने यही समझा था कि प्लाज्मा कॉन्सेंट्रेशन, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन कब बढ़ती है? जब लॉस ऑफ ECF वॉल्यूम होता है। तो वही यहाँ हो रहा है, इसीलिए नाउ यू नो द बेसिस, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन यहाँ बढ़ जाएगी बिकॉज ऑफ लॉस ऑफ वॉल्यूम। और हेमाटोक्रिट, बाय द सेम लॉजिक यू वुड एक्सपेक्ट कि हेमाटोक्रिट भी बढ़ेगा, राइट? अगर आप आइसोऑस्मोटिक को लेके चल रहे हो तो यू वुड एक्सपेक्ट कि हेमाटोक्रिट भी बढ़ेगा, बट although expected so, it remains unchanged because water shifts out of the RBCs, decreasing their volume and offsetting the concentrating effect of decreased ECF volume. ठीक है, सो एक ये थोड़ा सा चेंज है यहाँ पे कि हेमाटोक्रिट दो एक्सपेक्टेड था बढ़ेगा बट वो अनचेंज रहता है बिकॉज आरबीसी से वाटर शिफ्ट आउट हो गया है जिससे उनका वॉल्यूम कम हो जाएगा. दैट विल ऑफसेट द कॉन्सेंट्रेटिंग इफेक्ट ऑफ डिक्रीजिंग ECF वॉल्यूम. ठीक है, आई होप इट्स क्लियर. एंड करेक्टिव रिस्पॉन्स क्या होगा? अब अभी हमने कहा कि ECF ऑस्मोलैलिटी बढ़ी हुई थी, राइट? तो यहीं पे आ जाओ वापस, ये आ गया ना आपका, यही मैकेनिज्म आ गया. ECF ऑस्मोलैलिटी बढ़ी हुई थी तो क्या होगा? आई थिंक नाउ दिस शुड बी लिटरली ऑन योर टिप्स, नहीं? दैट स्टिमुलेशन ऑफ ऑस्मोरिसेप्टर्स, ठीक है, एंड this whole process will again work and restore your plasma osmolality and plasma volume to normal level. ठीक है, so this is what happens in the fourth situation. अब आगे बढ़ते हैं, लास्ट टू रह गई. हाइपोऑस्मोटिक वॉल्यूम एक्सपेंशन. ठीक है, ये ऑब्जर्व करते रहो कि किस कंपार्टमेंट में क्या चेंज आ रहा है, कीपिंग द y and x axis in mind, है ना, ये वॉल्यूम और ये ऑस्मोलैरिटी. सो हाइपोऑस्मोटिक वॉल्यूम एक्सपेंशन पहले तो होती किन केसेस में है? ड्यू टू गेन ऑफ वाटर इन कंडीशंस लाइक सिंड्रोम ऑफ अप्रोप्रिएट ADH सिक्रीशन, जिसको हम SAIDH बोलते हैं, ingestion of large volume of water, or if there is rectocolonic washouts with plain water. ठीक है, अब इस सिचुएशन में क्या होगा? Look at the y-axis. ठीक है, there is some change here in osmolality of both the compartments if you see. So इस केस में ECF osmolality कम हो गई because of excess water retention. ठीक है, तो क्या हुआ? ECF वॉल्यूम जो है बढ़ गया है बिकॉज ऑफ एक्सेस वाटर, और उसी एक्सेस वाटर की वजह से ECF की ये ऑस्मोलैलिटी यहां से देखो कम हो गई है, 300 से कम हो गई है, राइट? अब उल्टा होगा, जो हमने अभी सिचुएशन अभी तक बात कर रहे थे ICF से ECF में शिफ्टिंग की, ऑपोजिट विल हैपन. नाउ वाटर विल शिफ्ट फ्रॉम ECF टू ICF. ठीक है, जिससे कि ICF का वॉल्यूम बढ़ गया, राइट? डू वी सी यहाँ पे, ये देखो इस एक्सेस में वॉल्यूम बढ़ गया है ना, दोनों कंपार्टमेंट्स का बढ़ा हुआ है. इस केस में ICF का भी बढ़ गया है, जो अभी तक हम बात नहीं कर रहे थे. और ICF का वॉल्यूम बढ़ गया है तो उसकी ऑस्मोलैलिटी कम हो जाएगी until it equals the osmolality of ECF, as has been happening in all the situations. अब इस केस में क्या लगता है आपको, प्लाज्मा प्रोटीन्स में क्या होगा? व्हाट डू यू थिंक? सो कोरिलेट करो शुरू से कि ECF वॉल्यूम बढ़ा है ना इसमें भी, तो प्लाज्मा कॉन्सेंट्रेशन, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन कम हो जाएगी. क्यों कम हो जाएगी? नाउ आई एम सेइंग इट फॉर द थर्ड टाइम, बिकॉज एडिशनल फ्लूइड से डाइल्यूशन होने की वजह से. ठीक है, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन कम हो जाती है उससे, और उससे प्लाज्मा कोलॉइड ऑस्मोटिक प्रेशर भी कम हो जाता है ना, ये हमने सबसे पहले बात की थी. और हेमाटोक्रिट इस केस में अगेन वी एक्सपेक्ट की कम हो जाएगा, जैसे हमने आइसोऑस्मोटिक सिचुएशन में देखा था. प्लाज्मा प्रोटीन यहाँ कम हो रहे थे और हेमाटोक्रिट भी कम हो रहा था, बट इसमें भी हेमाटोक्रिट एक्चुअली अनचेंज रहता है, बिकॉज इस केस में जो हमने यहाँ रीजनिंग दी वही रीजनिंग यहाँ है कि बिकॉज वाटर अब इट शिफ्ट्स इनटू द आरबीसीज, increasing their volume and offsetting the diluting effect of the gains of ECF volume. ठीक है, तो इन दो सिचुएशंस में आपके हेमाटोक्रिट का थोड़ा अनएक्सपेक्टेड चेंज, मतलब अनएक्सपेक्टेड सिचुएशन है कि इट इज एक्चुअली रिमेनिंग अनचेंज इन दोनों में। ठीक है, यहाँ तो डिक्रीज इंक्रीज डिक्रीज आ रहा था जैसे हमें लग रहा था ट्रेंड में, बट इन दोनों में हमारा हेमाटोक्रिट अनचेंज रहा, क्योंकि आरबीसीज में एडिशन या सब्ट्रैक्शन ऑफ वाटर हो रही है, जिससे आपके कॉन्सेंट्रेटिंग इफेक्ट ऑफ कम ऑफ डिक्रीज ईसीएफ एंड डाइल्यूटिंग इफेक्ट ऑफ मोर ईसीएफ, वो दोनों मैकेनिज्म्स ऑफसेट हो रहे हैं इस वजह से। ठीक है, करेक्टिव रिस्पॉन्स क्या होगा इस सिचुएशन में? जो वैस्कुलर वॉल्यूम रिसेप्टर्स हैं और ऑस्मो रिसेप्टर्स, they sense the changes in plasma volume and osmolality, right, as they have been doing. So signals from either of them will lead to excretion of large amount of hypotonic urine, जिसको हम कहते हैं water diuresis, जो हमने last time पढ़ा था, which gradually restores the plasma volume and osmolality to normal. ठीक है, is this clear? I hope it is, because then we can move on to the last situation here, the last disturbance which can occur, which is a hypoosmotic volume contraction. ठीक है, ये किस situation में होती है? When there is loss of sodium chloride or hypotonic fluid from the body. ठीक है, ये कब होता है? This can occur in a adrenocortical insufficiency, ठीक है, which is associated with renal loss of sodium chloride, or this can occur in case of vomiting or aspiration of gastric secretions, जो होता है associated with hypertonic fluid loss from the body. ठीक है, now let's learn the consequences. So again आप देखो कि यहाँ पे जहाँ osmolality बड़ी थी इन दोनों में, अगर आप बस डायग्राम और एक्सिस दोनों को ऑब्जर्व करते रहोगे तो यू विल नॉट फॉरगेट। इन दोनों में यहाँ देखो बढ़ गई थी ना, और यहाँ पे दोनों में वाई एक्सिस आपका घट गया है, एक्स भी घटा है इन दिस केस। सो इसमें क्या हुआ? अगेन ECF ऑस्मोलालिटी जो है कम हो रही है, बिकॉज देयर इज लॉस ऑफ सोडियम क्लोराइड एंड अदर सोल्यूट्स, है ना। हमने अभी बात की डायरिया, वॉमिटिंग और इन सब सिचुएशंस की, तो देयर इज लॉस ऑफ सोडियम क्लोराइड एंड अदर सोल्यूट्स। नाउ अगेन वाटर विल शिफ्ट फ्रॉम ECF टू ICF। ठीक है, उससे क्या होता है ना, आई थिंक दिस शुड बी ऑन योर टिप्स लिटरली। ICF का वॉल्यूम बढ़ गया, और ECF का वॉल्यूम कम हो गया। ठीक है, पहले क्यों बढ़ा था ECF का? क्योंकि एक्सेस वाटर रिटेंशन हो गई थी, राइट। इस केस में तो वाटर और सोल्यूट लॉस हो रहा है, राइट। सो ECF वॉल्यूम आपका कम हो गया। अब ICF में वॉल्यूम बढ़ गया तो ऑफ कोर्स वी नो क्या होगा, ICF ऑस्मोलालिटी कम हो जाएगी until it will equal ECF osmolality, right? Now अब जब प्लाज्मा वॉल्यूम और ऑस्मोलालिटी चेंजेस जो होते हैं, दे टेंड टू पैरेलल दोज ऑफ द ECF। ठीक है, however a decrease in plasma volume raises plasma protein concentration। ठीक है, अभी पिछले सिचुएशन में क्या हुआ था, प्लाज्मा प्रोटीन कॉन्सेंट्रेशन कम हो गई थी, बट अभी हमारा प्लाज्मा वॉल्यूम कम हो गया है, तो प्लाज्मा प्रोटीन कॉन्सेंट्रेशन बढ़ गई। वी नो उससे क्या होता है, ऑनकोटिक प्रेशर बढ़ जाएगा, राइट। बिकॉज ऑफ दिस रेज्ड ऑनकोटिक प्रेशर, शिफ्ट ऑफ फ्लूइड फ्रॉम द प्लाज्मा टू इंटरस्टिशियल फ्लूइड इज डिक्रीज्ड। ठीक है, so because of this plasma volume is better preserved than the volume of interstitial fluid। ठीक है, because oncotic pressure ने क्या किया, shift of fluid कर दिया from plasma to interstitial प्लाज्मा फ्लूइड से इंटरस्टिशियल फ्लूइड में कम कर दिया शिफ्ट ऑफ फ्लूइड, सो प्लाज्मा वॉल्यूम आपका बेटर प्रिजर्व हो गया देन द वॉल्यूम ऑफ इंटरस्टिशियल फ्लूइड. But in severe cases like a grave salt deficiency syndrome, the plasma volume reduces sufficiently to produce a shock-like state with signs of renal failure. But unlike in shock, there is no sensation of thirst. जो इसमें होता है. ऐसा क्यों होता है? Because आपके cells of thirst center जो हैं वो swell up हो जाते हैं due to shift of water from the ECF to ICF. ठीक है. जो thirst center है that interprets the swelling of the cells as presence of excess water. तो उससे thirst inhibit हो जाती है क्योंकि वो interpret ही उन cells की swelling को ऐसे करता है कि there is excess water in the body, which is not the case, right? तो thirst inhibit कर देता है. और जो hematocrit है वो बढ़ जाएगा because ECF वॉल्यूम तो कम हो गया है और RBCs जो आपके हैं they will swell because of water entry. ब्लड प्रेशर को क्या होगा? It will decrease because of decrease in ECF volume. Remember इसमें हमने we have talked about vomiting, aspiration of gastric secretions. Okay, we are talking about hypertonic fluid loss. ठीक है, तो BP तो affect होगा ही, in fact कम हो जाएगा because ECF volume कम हो गया. करेक्टिव रिस्पॉन्स क्या होगा? ECF वॉल्यूम कम था, राइट, बट थर्स्ट तो एब्सेंट है, राइट. सो दिस इज मोर लाइक अ नेचर्स डिफेंस मैकेनिज्म. ठीक है, कि लुक एट इट लाइक दिस कि अगर कोई भी स्टेज पे वाटर इंजेस्ट होता है दैट विल प्रॉम्प्टली लीव द ECF एंड एक्सपैंड द ICF इवन मोर. ठीक है, सो सॉल्ट क्रेविंग या सॉल्ट एपेटाइट जिससे इंडिविजुअल का मन करेगा कि वो और ज्यादा अमाउंट ऑफ सोडियम क्लोराइड कंज्यूम करे, दैट विल हेल्प इन रिस्टोरिंग ECF osmolality. ठीक है, सो normalization of ECF osmolality that will shift your water from ICF to ECF, leading to partial restoration of ECF volume shift of fluid. ठीक है, from ICF to ECF, that will shrink the cells of thirst center and ultimately आपका thirst restore हो जाएगी. ठीक है, so consequently when drinking of water will occur because of restoration of thirst mechanism, तो जो remaining deficit है ECF volume का that will be made up for. ठीक है, तो ये थोड़ा सा डिफरेंट फंडा है इन दिस लास्ट वन, हाइपोऑस्मोटिक वॉल्यूम कॉन्ट्रैक्शन. जो याद रखने वाली चीज है कि इस केस में थर्स्ट मैकेनिज्म इनहिबिट हो जाता है बिकॉज इट्स इज बॉडीज वे ऑफ इंश्योरिंग कि जो भी उस वक्त वाटर इंजेस्ट है वो प्रॉम्प्टली चले जाएगा और इनफैक्ट ICF और एक्सपैंड हो जाएगा उस चक्कर में. सो इसलिए क्या होता है कि सॉल्ट क्रेविंग होती है इंडिविजुअल को जिससे वो सोडियम क्लोराइड टाइप सोल्यूशन कंज्यूम करने की कोशिश करेगा इंडिविजुअल. उससे आपकी ECF ऑस्मोलैलिटी जो कम हुई हुई थी वो रिस्टोर होनी शुरू हो जाएगी. जैसे ही वो नॉर्मलाइज होनी शुरू होएगी, ठीक है, उससे वाटर जो शिफ्ट होना शुरू हो जाएगा फ्रॉम ICF टू ECF, उल्टा होना शुरू हो जाएगा, कॉन्ट्ररी टू व्हाट हैड हैपेंड. उससे क्या होगा? पार्शियल रेस्टोरेशन तो हो जाएगी ECF वॉल्यूम की, है ना. उससे जो सेल्स हम कह रहे थे कि क्यों थर्स्ट सेंटर इनहिबिट हुआ था, क्योंकि वहां स्वेलिंग हो गई थी सेल्स की बिकॉज ऑफ शिफ्ट अर्लियर फ्रॉम ECF टू ICF. अब उस एरिया में उन सेल्स की श्रिंकिंग हो जाएगी जिससे आपका थर्स्ट सेंटर स्टिमुलेट हो जाएगा. ठीक है, इट विल रिस्टोर द थर्स्ट. तो अब जब वाटर कंज्यूम करेगा इंडिविजुअल तो जो भी अब डेफिसिट रह गया था ECF में दैट विल बी मेड अप फॉर. Okay, I hope this is a bit exhaustive, I know, but this is important. And I hope you all have got some clarity. When you sit down, study this diagram along with the tables that I'm going to show you next. They summarize everything that I just spoke. I don't think you'll forget it. I think, in fact, the concepts will become very strong. So remember, this is called as the Darrow-Yannet diagram. And this is what I just told you about. Just go through this table once. इसमें सारा कुछ दिया है of each disturbance. The causes, as I explained to you, the causes of each, consequences of each, and corrective response of each in points. So this will really make it easy for you to recall. I'll give you a bit of time to just study this. And there are, I mean, this is the part 2 of it. ये सारे आपके dehydration के disturbances थे, right? और ये सारे आपके overhydration के हैं. Have a look, just go through. ठीक है, so this is all for today. If you want, I can go through this again, although I think I've said everything that there is in these tables. इसमें बहुत अच्छे से summarize हो गया है. So anyway, are there any doubts or queries? Let me know, or else we can call it a day. Yes, anyone? Okay, I don't see any raised hands on my screen. So feel free to speak up if you have anything to ask me. Otherwise, of course, as Gauri has told me, you all prefer to, I think, go through the portion once before coming up with any doubts and queries. So yes, you can do that and forward them to Gauri, and she will send them to me. I'll be happy to answer them. Okay, so I'm still not getting anything regarding this. Shall we stop the session then? Shall we end today's lecture? Okay, so that's all for today then. As I said, you can forward your queries to me. So thank you very much, and I hope you have a great day.